Hi, it's Mr. Anderson, and in this video I'm going to go through chromosomal inheritance. Most students understand Mendelian genetics and could do a simple Punnett square like this, but in this video we're going to dig more deeply and see that those genes are actually carried on chromosomes and packaged into gametes. And so it's built on the work of Thomas Hunt Morgan. And so we're going to go through a monohybrid and a dihybrid cross, not only the Punnett square, but let's dig into how the chromosomes are actually delivering those genes. And so in this monohybrid cross, this first Mendelian cross, he crossed, remember, yellow with green and got just yellow seeds. And then he selfed yellow with themselves and found that 3-to-1 ratio. So if we were to add letters to that represent the genes, the parents are going to be homozygous dominant and recessive. In the F1 generation, they're going to be heterozygous. And then we produce a Punnett square that shows us all the alternatives that we could get from that F1 cross. And so what do these two sides really represent? This big Y represents the odds of delivering that big Y allele or that dominant allele. And this is the odds of representing that little allele or that recessive allele. And so there's a 50% probability. And so these are really the odds of producing gametes that have the dominant gene or the recessive gene. But remember, you don't just produce 2 gametes, you produce 4. And so we're going to dig into that in just a little bit. These would be the gametes from the other offspring or the other parent, and then these are going to be the 4 possible offspring that we could get. Now remember, those genes are actually delivered on chromosomes. And so if we look at it at the level of Thomas Hunt Morgan, and in this model I just have 2 chromosomes here, we find that that dominant gene is actually found on a chromosome, and the recessive gene is found on a homologous chromosome. In other words, it's the same length. Now, where did these chromosomes come from? They came from the P generation. And so in the P generation, you could imagine that that parent would have 4 chromosomes. They'd all be red, and each of their chromosomes, too, are going to have those dominant Y genes, and likewise for the other parent. And so now we have to figure out what possible gametes could come from this. And so we have to dig into meiosis. Remember, meiosis starts with duplication of the DNA and then 2 divisions. And so let's duplicate the DNA and see what happens. We've got 2 that have the big Y and then 2 that have the little y. And so you could imagine we're going to make 4 gametes, and you can almost visualize those. So each of these vertically is going to make a gamete. And so how many possibilities do you see so far? I just see the big Y, here's another big Y, or the little y. Now remember, during meiosis, they're going to independently orient themselves at the metaphase plate. So they could line up like that, or they could line up like that. Or they could line up like that, or they could line up like that. There are 4 arrangements. But it doesn't really matter how they're orienting themselves. We're still seeing those 4 possible gametes, but 2 of them are going to be exactly the same. Each sex cell is either going to get a big Y or it's going to get a little y. And so let's see those 2 divisions in meiosis. So there's one division and there's another division. So remember, you produce 4 cells in meiosis, but you can see that 2 of those are going to be the exact same genes, the exact same chromosomes. And so we really only have 2 alternatives, big Y or little y. Now, how does fertilization work? Remember, we're going to have those gametes of the other parent, and then they're going to combine. And since there's 2 possibilities on each of the parent, we could only have 4 possibilities of the offspring. Let's kind of visualize those. We could get a big Y from each of the parent. So that'd be one possible zygote, and that's going to be a yellow seed. We could arrange it like this, get a dominant from one, a recessive from the other, or a dominant from this one and a recessive from the other. Those are each going to be yellow as well. Or we could get 2 recessive genes or recessive chromosomes. And so how many possibilities do we get? We get a 3-to-1. Now the Punnett square worked great. We didn't learn anything new really by looking at the level of the chromosome. That's about to change. Now let's look at a dihybrid cross. And so in this cross, Mendel crossed yellow round with green wrinkled, and he found in the F1 generation that they were all yellow round. So yellow and round seem to be dominant. And so if we were to add letters to that, these would be homozygous, yellow, yellow, round, round versus green, green, wrinkled, wrinkled. And so what's going to be our F1 generation? They're each giving one of each of those letters, so they're going to be hybrid for both. But now Mendel's presented with this problem. And so do those genes travel together? Are they dependent on one another, or are they independent of one another? Because those 2 alternatives could produce different possibilities. You could get a Punnett square that looked like this, where we only have 2 possible phenotypes, or we could have 4 possible phenotypes and tons of different ratios. And so you probably know what's the right answer, but let's go through the Morgan way. Look at the chromosomes and figure out what's going on with those genes. And so if we go back to the F1 generation, and let's just put the yellow and the green on our respective chromosomes, now the question is begging, is the R gene, is that round gene, going to be found on the same chromosome as the yellow, or is it going to be found on a different chromosome? And so let's just play out those two possibilities. Let's just say that the round is found on the same chromosome. So let's put it right here. Again, you're getting the round and the wrinkled gene from that one parent, the dominant parent, we could say, and then the two recessives from the recessive parent. And so let's go through all the possibilities that we could get. And so now we've got interphase where we copy the chromosomes. And now we have to figure out all those different orientations. So it could line up like this. This would be a gamete that's going to be round and yellow. And this would be a gamete that's going to be green and wrinkled. And so let's figure out if they orient themselves in a different way. Does that change the different gametes that we could get? So they could line up like that. That's not really changing the possibilities. They could line up like that. It's not really changing anything. You can see that the round is going with the yellow and the wrinkled is going with the green. They could orient themselves like that as well. And so really we only get two possible gametes in this case. And so if we were to go through meiosis, let's grab one like that. This would be one parent. This would be the other parent. So how many possibilities we could get? Well, since this parent only has two gametes and this does as well, there's really only 4 boxes in our Punnett square. So you could have an individual made from round and yellow, so dominant for both traits. You could get a dominant from this side and a recessive from the other, or vice versa. Or we could get both of those recessive genes and it would look like that. So if we were to do this one alternate reality, how many possible phenotypes we would get? These are all going to be round yellow, except 1/4 of them are going to be wrinkled and green. And when Mendel did that cross, that didn't happen to be. And so we now know that those are going to be found on different chromosomes. And so the round and the yellow are found on different chromosomes. So let's go through meiosis and figure out how we're getting all these different possibilities. And so again, during interphase, they're going to copy all of their DNA. So we've duplicated the chromosomes. And so they could line up like this. And so what kind of a gamete would this be? It's going to be a round with a yellow and a wrinkled with a green. But if it lines up like that, nothing changes. We've still got round with yellow and wrinkled with green. But let's say it lines up like that. Now what do we have? You can visualize now we've got a wrinkled with a yellow, and we're also getting a round with a green. And so how many gamete possibilities do we have here? 4 possibilities. This again, 3 and 4 are going to be the exact same thing. And so now there are 4 possible gametes. And so if we were to play that out, using just a Punnett square, we'd have to put all those possibilities on one side, all those possibilities on another, and we'd find that all these individuals are going to be round and yellow. These 3 individuals are going to be round and green. These 3 are going to be wrinkled and yellow, and only one of those 16 possibilities are going to be wrinkled and green. Now, Mendel was able to figure this out, not understanding what's going on with chromosomes, and so it took decades before scientists looked at this and found a flaw in his model. And let me show you that. So in this Thomas Hunt Morgan cross, we're dealing now with fruit flies rather than peas. But in this cross, it's a simple test cross. So what he has is a wild fruit fly, but it's hybrid. So what does that mean? It has one of the wild genes, and this is for normal wings, with a vestigial gene, which would normally create these small little wings. And then it's also hybrid for coloration. So it's got the normal coloration, and then it has one of the genes for this black coloration. Likewise, this other parent over here is going to be recessive for both. And so when Thomas Hunt Morgan, because he understood Mendelian genetics, set up his Punnett square, he set it up like this. And so what are going to be the possible gametes that this fruit fly could give? Well, it could give VG plus, and it also could give the B plus, so it could give that. It could also give each of the recessive genes, so it could give that recessive vestigial wing gene and the black gene. It could also then give a combination of those two. So there are 4 possibilities that we could get from that one parent. Since this one is recessive for both, it really can only give one gamete. And so Morgan knew that there would only be 4 possibilities, and we should get 25% of each of these. So if you get these gametes, you should get a hybrid for each. And so you're going to look exactly like this parent. You're going to be wild type for both. If you're this, you're going to get all recessive genes, and so you're going to look like this parent. And so we call those parental phenotypes because they look just like the parent. This one here looks like that parent. This one looks like that parent right here. You could also get what are called recombinants. And so this one right here is going to have the normal wings, but it's going to have that black coloration. And this one over here is going to have normal coloration, but it's going to have those vestigial wings. And so Morgan knew that it would be 50% parental types, 50% recombinants. But when he actually did the cross, that's not the number he got. What he got was 17% recombinant, 83%. So Mendel was wrong, or his model didn't go deeply enough. And so in the next video on genetic recombination and gene mapping, I'm going to answer that and show you not only did Morgan figure out now that genes are carried on chromosomes, but the importance of crossing over, producing those recombinants. And so that's chromosomal inheritance. It's understanding genetics not at the level of Mendel, but also at the level of Morgan, and I hope that was helpful.